Hello and welcome to Regents Science Live. Thank you for being here tonight. My name is Lori Hennessy and I'm a living environment teacher at New Hartford High School. The goal tonight is to go over a few of the living environment topics that you're going to see tomorrow when you take the exam so that you feel a little bit more comfortable with them going into tomorrow. Before we get started, I just want to thank OHM BOCES for putting this on. I want to thank Joe Lynch and Proctor High School for hosting us here tonight at this facility. And I want to invite anyone that's watching tonight to either tweet in your questions. You can see the Twitter handle on your screen, or you could call in any questions that you have. We have the phone number listed on your screen. And while we wait for a few of those first questions to start coming in, I wanted to give you a little bit of an overview of some key concept ideas that we see every year on the Regents exam. These are guaranteed topics that are going to be worth points tomorrow. So if you want to hold a couple of questions, grab paper and pen or pencil and kind of jot down some notes. This is going to be some valuable information for you to know for tomorrow. All right, so on the screen here, you can see that these are going to be guaranteed topics on the Regents exam tomorrow. I went through and I tallied up some of the points from last year's Regents exam, and 9 total points out of 85 came from the topic of human impact on the environment. So I've got 2 bullets listed here, both negative and positive. Some negative impacts that we're going to start talking about first are climate change and carbon dioxide levels. What I would note is that carbon dioxide, this is the greenhouse gas. And various human activities are going to release carbon dioxide into the atmosphere. So that's going to increase carbon dioxide levels. Some human actions have led to the destruction of natural habitats, such as deforestation, removal of natural habitats for human activities, the use of finite or limited resources. This term, I feel like I've been seeing more and more. Okay, finite, it's kind of like the opposite of infinite, okay, so a limited resource. We've been introducing and also unknowingly or knowingly spreading invasive species around New York State. So these are non-native species that have been introduced into a habitat. Their population grows exponentially, and they can really take over that natural habitat and exploit the resources there, whether it be kind of like block the sunlight for native plants or kind of like decimate a whole population that was prey for another organism. The positive impacts of this are things like recycling, sustainable development, so, putting back a little bit of what we take, having an interest in renewable energy and kind of exploring options like that, introducing biological controls, which are things like introducing a predator to a pest that does belong in that ecosystem that could prey upon the pest and kind of control those population levels. Okay. And one thing is a trade-off, and this is a concept that's kind of abstract. Okay. Whenever you have one impact, there's always an opposite. So there's always going to be positive and negatives, like pros and cons, to all of these aspects. So something like renewable energy. Okay. It sounds like it's a positive. Think about some negative implications, whether it be cost or infrastructure, things like that, having to switch over. Those are some negative details that we have to deal with while we kind of make the move towards renewable energy. The second topic that I have 9 points on last year's Regents exam. The reason why this is kind of like, I guess, inflated to 9 is that because I counted everything where you were given data, either in a data table or a diagram, food web, some sort of picture, and you had to come to a conclusion based on what you saw in front of you. Okay, so not really you knowing a lot of outside information, outside sources, but looking what you have in front of you and coming to a conclusion. So that's kind of more of a skill that you need to have been, practicing rather than something that you can just, learn, memorize, and be able to do. Okay. The next one I want to go over with you, hypothesis. Independent variable and dependent variable, that's what those abbreviations stand for that I have listed there. So a hypothesis is an educated prediction as to what you think is going to happen in a controlled experiment. So it's not a question. It's, if as the scientist this action is taken, then we believe this will be the outcome. So it's always a statement, ends with a period. Okay, it relates right back to the experiment at hand, and it can be correct or incorrect. This is done at the beginning of an experiment to kind of like identify your goal, and from there, data is either going to support or refute the hypothesis. The independent variable, remember the I, okay? I can change the independent variable. The letter I looks like the number 1. Okay, the scientist can change one variable within an experiment at a time, and the independent variable is kind of like reflected in the dependent variable. Okay, dependent variable, remember the letter D? This stands for data. So what are the numbers that you're collecting in an experiment? Are you measuring time in seconds, plant growth in centimeters, okay, pulse rate, in beats per minute, things like that? You have to be able to identify a control group and state the importance of the control group. There's always 2 groups within an experiment. The control group is the first one I'm going to talk about. This group is going to show us kind of what happens in this experiment if you change nothing, if it's just normal conditions, whatever is normal for this organism, normal temperature if we're talking about bacteria, okay, normal food. Okay, if we're talking about plants, normal soil in terms of fertilizer or amount of water, things like that, whatever is normal. This is going to give us a baseline for comparison. Okay. And what we're going to compare to this is the results from the experimental group. The experimental group is going to be the same as the control group except for the independent variable. That will be changed, and you're going to collect data, compare your results, and see if your changes in the experimental group is significant compared to the control group. Okay. So being able to calculate an average. Okay, adding all of, a group together, dividing by the number that you have there, comes up with your average. That's an important way in order to compare 2 groups. Okay, make sure the sample size is large enough. If we're talking about an experiment, 5 is not a large enough number. Okay, if I was to experiment on myself with the number of clothespin squeezes that I can do in a minute, 1 individual is not a large enough sample size. Okay, so either increase the sample size, run the experiment again. That's going to give you more data and make your experiment more valid. All right. The third topic that I want to go over is energy flow through an ecosystem. This was 6 points on the June Regents. You need to know the types of nutrition. There's 2 categories. In autotrophic nutrition, these organisms are able to synthesize their own food. Okay. In heterotrophic nutrition, these organisms must find a way to obtain food or consume food. This relates right to the trophic levels. So the vocab trophic level is just each feeding level that you're going to see at each of these stages, whether it be like a food chain, food web, or a pyramid like this. The producers are always at the bottom. They're going to be letter A. They're autotrophic. They can synthesize their own food through the process of photosynthesis. And going up from that, you've got the primary consumers, which eat the producers. Those are herbivores. The secondary consumers, consuming the herbivores, they could be omnivores. All the way up, letter D could be the decomposers at the top. They're going to break down and recycle the nutrients back into the environment. Okay, so being able to read a food web is important. Lastly, there are questions about the 3 types of symbiosis. Mutualism and parasitism are kind of like the most common ones that we're going to see. In mutualism, you have 2 organisms that provide one another with a benefit. And in parasitism, you're going to see that one benefits and one is harmed. So typically the parasite, like think of a tick or a tapeworm, benefits from obtaining nutrients, whereas the host is harmed because they're losing that. They could, parasites spread diseases and things like that. With mutualism, you can think of pollination being a benefit to flowers and bees obtaining the nutrients from the flowers. All right, number 4 is immunity. The immune system is huge. Vaccines, kind of like really relevant for today, being able to give us an accurate definition of a vaccine. So what that would look like would be a weakened, a dead, or a part, like a section of RNA, something like that, piece of a virus that's introduced into the body. That would be the antigen. Remember, the G kind of like stands for germs. Your body produces antibodies. This is a big vocab word. Okay. And just kind of like knowing those 2 terms, antigen, antibody, and being able to kind of like differentiate that against an antibiotic. An antibiotic is a medication. You get prescribed that from a doctor. Okay, so nothing that the body produces or anything like that. That would be kind of like a trick question or a trick answer. Okay, it's involved with the immune system, but it's nothing that the body is doing in a response. Okay. The white blood cell is the part of the immune system that produces the antibodies, all right, that respond to the pathogens. Okay. Know the difference between allergies and viruses. The virus that is kind of like our, catch-all virus that is usually asked about is HIV and its impact on the immune system. It's going to kind of weaken the immune system. Okay, target the white blood cells so that the individual's immune response to any subsequent infection is going to be a lot lower or delayed, and so that individual could get a lot sicker. Okay. Across all body systems, I'm going to talk about one more body system, all right, but across all of them, the goal is to maintain homeostasis. So that's like our main theme. It's kind of like our catchphrase for this. All right, number 5. All right, natural selection, this was 5 points on the June Regents. Here I have an evolutionary pathway. It's called a cladogram. This is going to show like a visual representation of how organisms are related and kind of like their evolutionary past. Okay, you can always identify where's the past, where's the present. Sometimes this will be turned on its side. It could be horizontal. Okay, but once you orient yourself, it's kind of like visually really easy to read in terms of the organisms that are most closely related are going to be the ones that are closer together on the pathway and split or diverged more recently in time. Okay, so you're going to have to, mark up your paper, draw a line over to identify where they have diverged in time. The two causes of variation is mutations and sexual reproduction. A mutation is any change in DNA, and it can be passed on to the next generation if the mutation occurs in a gamete, which is a sex cell. Sexual reproduction, the process of meiosis, genetic recombination, and two parents combining their genetic information to create offspring is another big way that genetic diversity or genetic variation is introduced into a population. Okay. So those organisms that are best adapted to that current environment are going to be able to survive. And if organisms are surviving for a longer period of time, they're going to have the chance to create more offspring, reproduce. And the frequency of those advantageous traits, that what makes them best adapted, that's going to increase in frequency in the next generation. The last thing you're going to be asked about is kind of like our catchphrase for evolution. This is common ancestry. The most accurate information that you can look at when you're trying to determine common ancestry is similarities in molecular evidence. So this is going to be similarity in DNA and proteins. At the end, we're going to talk about a couple of tests that you can perform in order to sequence the DNA, different pigments or proteins to identify which organisms are most closely related. Before that, structural evidence can be collected, things like fossil evidence, looking under a microscope to view different parts of an organism, or just physically looking at the body plan, body shape, and features of different organisms to determine if they're related or not. All right, the two cell processes are photosynthesis and cell respiration. 5 points were given on the last Regents asked about these two processes. Okay, so I'm going to copy down the equation for photosynthesis. Plants take in carbon dioxide. They need water and to be in a place where they can receive the right amount of sunlight. And what they produce is oxygen gas and glucose. And water is also, on both sides, a waste product. The equation for cellular respiration, some people like to think about it as kind of like the opposite of this, opposite of photosynthesis. Okay, cellular respiration is the process that produces energy. And in order to gain the energy, you need to break glucose apart, okay, using oxygen gas and enzymes. Okay, that'll make the chemical reaction happen. And the waste products are carbon dioxide, water, and energy, ATP. Related to photosynthesis, knowing specialized plant cell structures in a leaf, the stomates or stomata, stoma, all of these variations of this word right here, they're found on the underside of leaves, the bottom of leaves, kind of like shielded from direct sunlight. Just like your skin has pores, there's openings in the leaf that are pores. Okay, they're regulated by two cells on either side, the guard cells. okay, that will open or close these stomates to let in carbon dioxide and to let out oxygen gas, okay, to let out extra water, things like that. Okay, the trick that you're going to be asked is what happens in plants. Okay, plants need to do both. All organisms, okay, plants, animals, fungi, bacteria, all animals need to perform cellular respiration, okay, because they need to make energy, ATP. So plants perform cellular respiration as well. They need to photosynthesize to make the glucose, whereas other organisms can obtain glucose through feeding, things like that. I told you I would talk about one more body system, and it's going to be the reproductive system. We're going to focus on the female reproductive system. This is the one that you need to know. A lot more happens with the female reproductive system, okay? So you need to know the parts and the functions. I feel like this picture would be a great picture to put on part C, to have it as a diagram, and then for you to answer some short answer questions about it, okay? So you're going to have to know what you are looking at, okay? So what we're looking at is letter C is an ovary. Okay. The function of the ovary is to release female hormones and also hold immature eggs. And once a month, an egg is released, okay, into this transport tube right here. It's labeled on the other side. Okay. This is called the oviduct. Or fallopian tube. And this is where fertilization takes place. If sperm is present, the sperm could meet the egg, fertilize, form a zygote, and that zygote would then implant into this structure here, which is letter D, the uterus. Okay, one important organ that is formed during pregnancy is the placenta, letter B. Okay. This is where diffusion takes place. Okay. Oxygen and nutrients, things that humans need to stay alive, that's going to diffuse from the mother to the fetus. And then waste products, like carbon dioxide gas, is going to diffuse from the fetus to the mother, okay, and then the mother will excrete the waste. Okay. Inside the placenta, there are these blood vessels called capillaries, so the mother's blood and the fetus's blood don't mix. They'll run past each other, and diffusion is the process that'll kind of take place. Okay. The two female hormones that you need to know are estrogen and progesterone. Okay. They both regulate this female reproductive cycle, and gametes for the female are egg. Okay, gametes for the male, sperm. Both of the nucleus in an egg and a sperm cell are haploid, and that means that they have half the number of chromosomes that the zygote is going to carry, because a half plus a half, equals a whole. So an egg plus a sperm equals a zygote, an embryo, a fetus, and then a newborn. All right, I'm going to go over one more topic, and then I'm going to kind of take any questions that you guys have had up until this point, any questions about these topics or something else. Make sure that you're sending them to us so that we can kind of go over and answer them with you. The last topic for right now is genetic engineering or gene editing. This was 2 points on last year's Regents. Okay, this diagram right here showing how to insert a gene that codes for an important human protein into a bacteria cell. Okay. So that bacteria cell can synthesize the human protein that can be purified and administered to humans. Okay. In addition, replacing any unwanted gene with one that is desirable from a species or a different species. Okay. And the protein that causes these reactions to occur are enzymes. Okay, restriction enzymes cut DNA, and other enzymes kind of act like glue, and they're going to kind of seal the DNA pieces together so that this can take place. Okay, genetic engineering, this might be a phrase that you're either going to have to recognize or kind of see in a multiple choice answer or something like that, and be able to pick that one when you see a diagram like this, or you see two different species genes being kind of combined in one organism. All right, so that was kind of a lot of content. That was a lot of points that you could have got on that last Regents. So I'm going to ask my people that I have here, are there any questions that you guys have for me from the people that are watching today? Yeah, all right, I'm going to invite Preston up. Come on up, you can ask me.
Our first question comes from Jack from Mount Sinai, New York. He wants to know what the difference between passive and active transport.
Okay, that's a good question. All right, so what I'm going to do is I'm going to go over it kind of on this sheet of paper here, okay, so that I can show you a diagram of what it looks like, passive versus active transport. Passive transport is going to be the movement of molecules from high to low concentration. These brackets, that's the kind of symbol for concentration, okay? So if you have a cell membrane, I'm going to draw it as kind of like this dotted line here because membranes are semi-permeable. They're going to allow some material to pass through while, not allowing others to as easily. Okay, high concentration, okay, would be crowded. Okay, many molecules on one side in an area compared to low concentration, maybe none or just a few. Okay, high to low, passive transport is going to be this way. Okay, this occurs without the use of energy. Okay, and kind of like almost a synonym for passive transport, but this would be kind of like a type of passive transport is diffusion. Okay. A specific movement called osmosis. This is going to be the diffusion of water. Okay. Water is going to move in the same manner, from high concentration to low concentration without energy. Okay. Compare that to active transport. Okay. The opposite definition is going to be the movement of molecules. Okay, from low to high. So if I draw you a visual for this, this is going to be your semi-permeable membrane. Okay, low concentration in the bloodstream, and then high concentration in a cell, like a muscle cell or a nerve cell. Okay, the body uses energy to pack these molecules into the cell. The best example are ions, like sodium ions or potassium ions, okay? Things with a charge that are going to be packed inside cells, okay? So that was a good question. We're going to come back to these two terms when we go over one of your state labs, all right, with one of these, okay? But I'll leave this up here, okay, for passive and active transport. Okay, what's next? Danielle, I see you. Come on over.
Hi, this is an anonymous question, and they're wondering, how does the digestive system work?
Okay, so the digestive system is going to be one of the body systems that are not as asked on the Regents exam. What I see the most is the regulation of blood sugar in relation to maintaining homeostasis. Okay, so after an individual eats, like if they eat something high in carbohydrates, the digestive system is going to break down the carbohydrates into simple sugars, like glucose is the best example of a simple sugar. Okay, and because of that, the body has to deal with that excess sugar. Okay, so what the body will do is the pancreas specifically is going to release insulin into the bloodstream. And that's going to shuttle the glucose into muscle cells or organs like the liver to decrease the sugar concentration in the bloodstream. That's going to restore it to normal. Okay, kind of like the opposite of that, if blood sugar is low, a different hormone will be released. It's called glucagon, and that will cause the release of stored glucose in the body. So like the liver stores a lot of sugar, it'll be broken down and released into the bloodstream to raise the blood sugar level. Okay, so when talking about the digestive system, knowing that specific example is a great way to explain how to maintain homeostasis. That's the best way to show this happening. Okay, insulin, pancreas, and regulating blood sugar level. That's what I would say to focus on for the digestive system specifically. Other organs and specific enzymes within the digestive system, like how amylase is an enzyme in the saliva, part of the mouth. Okay, that's kind of beyond the scope. We're more picky than what this Regents exam is going to ask. Okay. All right, that was a good one. Okay. Yeah, you can go.
Our next question is from Tati from New Hartford, and
Okay.
she wants to know what are the levels of organization within an organism?
Okay, Tati, that's a good question. So I'm going to go back to my paper here because that relates right back to kind of, the human body systems that we've been talking about, the levels of organization. Okay. The smallest unit in the human body is cells, the basic unit. Okay. A group of cells that work together to perform kind of like a common function, those are going to be called tissues. Okay. A group of tissues working together. Okay. Those are going to make up the different organs. A group of organs that work together to kind of achieve a common goal. Okay, our organ systems, and then a bunch of organ systems kind of put together. Okay, we'll give you the organism. Okay, this, I can guarantee it. Okay, we can all thank Tati tomorrow when we get this right and we all get a point. It's either going to be a multiple choice question or it will be kind of fill in the blank. So organ system is going to be omitted. Maybe cells is going to be omitted, and you're going to have to come up with that term and write it in a blank. Okay, so thank you. All right, Danielle, you're up.
Hi, I have a question from Rose, and it says, can you go over ecological succession and symbiosis?
Yes. Yep. So I'm going to go back to, okay. All right, got to get my bearings. So symbiosis. Symbiosis means a close relationship. So when I say close, I mean like literally on top of or inside of another organism. All right, there are three types of symbiosis. Mutualism is when both organisms benefit. Okay, commensalism is when one organism benefits and the other one is not affected at all. Okay, so usually that's abbreviated kind of like +0, okay, not benefited at all. Okay. And then lastly, parasitism, the host is harmed, that's my negative, and the parasite, like a tick or something like that, is going to benefit. Okay, so a symbiotic relationship is a close relationship. Related in the unit of ecology is ecological succession. Okay. Ecological succession is the orderly series of changes in an environment after a disturbance. So something like a forest fire or an abandoned farmland or any sort of like abandoned building, things like that, okay, that would start ecological succession. Over time and a long period of time, okay, the ecosystem would maintain its, or would come back to its natural state. So in this area, we kind of live in like this deciduous forest kind of woodland. So we would get grasses, shrubs, small conifer trees, which are evergreen trees, and then larger hardwood trees, okay, and eventually a climax community, which is the final state, that's going to be the forest, is going to be created. And that climax community is going to exist because it has high biodiversity, it's stable, until another either natural disaster or human interference comes through and kind of starts this process of ecological succession over again. So those two, related in ecology, those were good questions. Jaden, yeah, I hear the phones kind of ringing. You have a lot for me, so let's hear it.
So we have, so our first anonymous one is, how does meiosis work?
Yeah.
How does the prophase, telophase, metaphase, and the anaphase and telekinesis work together?
Okay, yeah, cytokinesis, not
Cytokinesis.
yeah.
Oh
no,
my
no,
God,
no.
I'm
Yeah,
sorry.
a lot of people say that, and I see that a lot,
Yeah.
so I know what you're saying. Yeah.
Okay.
So those specific phases of meiosis, prophase, metaphase, anaphase, telophase 1, and then we do it all over again for the second phase. That's kind of like beyond the scope of what's going to be on the Regents exam and assessed. But the general kind of broad overview of meiosis, that is something important to know. So meiosis is how I always pronounce it, like I really enunciate the E. It should be pronounced meiosis, but you got to remember the E because that stands for sex cells, okay? Sex cells are gametes. The two gametes, male is sperm and female is egg. Okay. The nucleus inside those two specialized cells are haploid. Okay, so haploid, remember the H, that stands for half. Okay, it contains one set of chromosomes, half, all right, of what the organism is going to have, right? We are diploid organisms, two parents, diploid means two. Okay, so we have two complete sets of chromosomes, genetic material. One half comes from one parent, the other half comes from the second parent, and the sperm and egg combine to form a zygote, and that is kind of like day one, first body cell. Okay, so those questions about meiosis or meiosis, remember the E. Okay, those are going to ask you, number one, what are the sex cells, sperm and egg, and haploid. Haploid nucleus is contained within each of them. Okay, two parents mixing their genetic information to create a new variety in the offspring. Okay, come on back up.
All right. So this one has a lot of questions as well. This
Okay.
is also anonymous.
Yep.
So the difference between photosynthesis and cellular respiration, the different microscope parts and the brief definition of them, cell organelles and a brief definition of them as
Okay,
well. All right.
yeah, thank you. Okay, so I feel like the microscope and the cell organelle questions go together. But I'm going to start with the photosynthesis and respiration equations. I'm going to go back to my sheet and flip to where I have them up, okay, right here, okay? So the photosynthesis equation is right here, okay? Inorganic molecules, carbon dioxide and water, okay, using light energy is going to create an organic molecule. When you see organic molecule, you should think glucose. Organic molecules, food, glucose, okay? Also, through the process of photosynthesis, oxygen is created. Photosynthesis is the process that decreases the amount of atmospheric carbon dioxide. Okay, the carbon is formatted into glucose and oxygen gas is released. Okay, cellular respiration, or I'm going to stop myself there, all right, because I'm going to relate this to the organelle question. Okay, an organelle that you need to know is the chloroplast. Okay, that's the site of photosynthesis. Okay. So before I finish talking about photosynthesis, I talked about guard cells and stoma, the opening in plants. I have an old Regents with me. This is what I'm talking about, okay? The stomate is the opening, these specialized cells, guard cells, okay? And if you take a look at question 29 for this, okay, it's essentially asking you about photosynthesis. Unfortunately, the answer that says photosynthesis is not the answer. Okay, the exchange of gases, carbon dioxide and oxygen, okay, is regulated by guard cells, these specialized cells on the underside of plants. Okay, so for cellular respiration, okay, I have the equation up here. Glucose, that organic molecule, think whenever you think organic molecule, think in your head glucose, plus oxygen gas, and enzymes are necessary to create carbon dioxide, water, okay, and ATP. I underlined ATP. ATP is energy. That's what you need to know, okay? Glucose gets broken down through a series of steps to release energy. The organelle that this occurs in is the mitochondria. Okay. All right. Before I move on and talk about the other organelles, main organelles that I think that you're going to see, I'm going to show this, okay? All right. So, what biological process? Your options are photosynthesis, respiration, are represented in the diagram below. Okay, so I'm looking at question number 28. I see green plant cell, and I really want to put photosynthesis, okay? That is my first thought, okay? Green, I think of photosynthesis because it's a plant, okay? But, you know, you can't just focus in on that. Food, so that's glucose, organic molecules. Okay, plus oxygen produces ATP, carbon dioxide, water. Okay, it's respiration. Okay, so that is the trick that I told you guys about. Okay, they're going to try and trick you. What do plants do? Both. Okay, photosynthesis and cell respiration. Okay, and on this page also, here's an ATP question. As for the parts of the microscope, I'm going to kind of skip over that. You know, there really isn't microscope questions if I'm thinking about, you know, points every year. If I'm going to guarantee you're going to see it tomorrow, I can't guarantee that you're going to see a parts of the microscope question. The Regents exam I have brought with me today, there aren't any pictures of the microscope. There is no microscope questions on this, but what can you see under a microscope? That was a good question. So here are the organelles, okay? Okay, so I'm going to skip mitochondria and chloroplast. Mitochondria is the site of cellular respiration, and the chloroplast is the site of photosynthesis, okay? The nucleus is the control center, where DNA is stored. DNA gets transcribed into mRNA, which is messenger RNA. It can leave the nucleus, travel through the cytoplasm, and I have that here. This is the jelly-like liquid inside of a cell, and that mRNA is going to end up in the ribosome. At the ribosome, a process called protein synthesis, okay, is going to happen. Where amino acids will be bonded together to create proteins. Okay. I told you I was going to skip both of these. Okay, the cell membrane is a semi-permeable membrane. Okay, that allows some material in and out. Okay, it's selectively permeable. So that's another phrase that you could see rather than semi-permeable is selectively permeable. So generally, we think about size, and small molecules are easily able to travel through the cell membrane. Okay, small. Okay. Whereas larger ones are blocked or have to enter and exit the cell kind of through a different avenue. Okay, maybe vacuole, right? I put this on here. This was like a stretch. Maybe you'll see it. Okay, definitely these. Maybe vacuole. All right, the vacuole is for storage. Okay, so I'll leave this up here. Center it a little bit better. Okay. All right, so those were good ones. Those were a lot. Okay. All right, where do we want to go next? Okay, back to Danielle. Thank you.
Hi, Sierra from Whitesboro is wondering the difference between antigens and antibodies and explanations for them.
Okay, yeah. All right, so an antibody is produced by white blood cells, okay, and they are specific to a disease like a virus or a bacteria that's present in the body, okay. So if the body is infected with a certain type of bacteria or a certain virus, your white blood cells are the part of the immune system. They're going to detect that infection. They're going to produce antibodies. And the antibodies are going to attack and destroy that virus or bacteria, okay? Each antibody is specific to the virus or bacteria that it's fighting for. So when I was little, if I had chicken pox, like you guys got vaccinated for chicken pox, we have the antibody for chicken pox in our bloodstream, okay? If we come into contact with another virus, that antibody isn't going to help us, right? We need to produce one that is specific to that virus, okay? So antibodies produced by white blood cells. That's a good vocab term to be able to write down tomorrow, not just recognize, okay? The immune system could be a Part C question, and knowing that antibodies produced by the body, the white blood cells, okay, to help fight off infections, okay? An antigen, remember the G? That stands for germ. That's a pathogen. That's a good word to know, okay? Okay, a pathogen is a virus or a bacteria, an antigen. Okay, so the antibodies, that's what they're looking for, the antigens. Anything that is foreign or unfamiliar to the body. Okay. All right, so those are the definitions. Part of the immune system, definitely something good to go over again. All right.
So I have two questions that go together.
Yep.
So we have, One from Natalie, who asks the stages of mitosis, and we have an anonymous question asking the definition of mitosis.
Okay, that's perfect. I'll go back and I'll say again the stages of mitosis, PMAT, prophase, metaphase, anaphase, telophase, knowing the specifics of what happens, where are the chromosomes, what is present or not, is kind of beyond the scope of the Regents exam. You're not going to be asked a question on when the nuclear membrane is going to break down in mitosis, okay? But the second question, kind of like the broad definition or the broad scope of mitosis, is that mitosis is going to create more body cells, okay? So mitosis, remember toe, it's part of your body, okay? Mitosis creates more body cells, and body cells are all cells in the body, so your skin cells, muscle cells, stomach cells, liver cells, okay? Everything except for sex cells, okay? Egg and sperm. Those are the only cells in the body that are not considered body cells, okay? So mitosis is the process that's going to produce body cells, okay? What is going to happen is the DNA inside the nucleus will be replicated or copied exactly. Okay. The nucleus is going to divide, and then the cell is going to divide into two. Okay. So mitosis is kind of a form of asexual reproduction. In the human body, it's how we create more body cells to create identical body cells to the original. And the overall purpose of mitosis is for growth and repair. So growth, like literally getting bigger, like getting taller, and repair. If I fall down and get a scrape, my skin cells surrounding that scrape are going to go through the process of mitosis, and they'll produce new skin cells to kind of seal up that cut, okay? So knowing what happens in the nucleus for both mitosis and meiosis, and the type of cell that is produced are kind of the two major themes when we talk about both of those processes, okay? Right, Preston's up.
Hennessy student Tati from New Hartford wants to know, what is the importance of autotrophic nutrition and decomposition?
Okay, that's a big one. I love it, okay? So I'm going to go back to my page here so I can write down some notes for you, okay? Knowing autotrophic nutrition, okay, that term, being able to write that term, being able to recognize that term is important. So auto, okay, means that you can do it by yourself, okay? And trophic is a feeding level. So autotrophic nutrition is when an organism can synthesize their own food. Synthesize means to make, and there are a couple of processes that'll do this. We need to know about photosynthesis, which I've gone over. So photosynthesis is the process and autotrophic is the type of nutrition. Okay. We think of photosynthesis as being the producers. Okay. Decomposers are generally at the end of a food chain or food web, and because decomposers break down and recycle nutrients back into the environment. So things like carbon or nitrogen, okay, can be broken down, break down dead organic matter like leaf litter on the forest floor. An organism that has died, okay, gets broken down and the nutrients get recycled back into the ecosystem. Two organisms that are great examples of decomposers that you might see in a food web are bacteria okay, and funguses, okay? So the two best examples, okay? Perfect. Thank you, Tati. Jaden's up. Yeah.
All right, we have a question from Jack from Clinton. He wants to know how DNA replication works.
Okay. So DNA replication, the word replicate means to copy. This is going to occur in the nucleus, okay? And when you copy the DNA bases, they make, an exact replica of that. So when I say the DNA bases, okay, those are the 4 letters that we talk about with DNA. The 4 bases are adenine, thymine, cytosine, and guanine. You don't need to know the names or be able to recognize those beyond the first letter. So A, T, C, and G. Okay. DNA is double-stranded. It's a double helix, so there's 2 sides to it. Okay. On one side, you're going to have a sequence of those nitrogen bases. The backbone is kind of the same on both sides. It's made up of sugar and phosphate. And what is important is the interior bases, the nitrogen bases, those A, T, C, G bases. Okay. When we replicate the DNA, the 2 strands of existing DNA are going to separate, and you're going to follow the base pairing rule in order to add a complementary strand or create that complementary strand. Okay. So A and T always go together. So always together, A and T. Okay. Whenever you see an A T bonds to it. Okay. A C and a G, cytosine and guanine, those are the other base pairs. Okay, so whenever you see a C, a G bonds to it. Okay, guanine bonds to cytosine. Okay, so those 4 nitrogen bases, AT bond together, CG bond together. Okay, that is going to be how the one strand of DNA splits and then makes the complementary strand to form 2 identical copies. Okay. Any change from the original is called a mutation. Okay, so a mutation, that's a good definition to know, any change in DNA. Okay, Danielle.
An anonymous question about synapsis.
Okay, synapsis is in one of the stages of meiosis. So again, that is beyond the scope of what you're going to be asked tomorrow on the Regents exam. It might be within a text or a question, but it's not going to be like the main part of it. But I will go over it. Synapsis occurs in prophase 1. All right, so in the first stage of meiosis, prophase, the homologous chromosomes, so all chromosomes have a matching pair, which is called a homologous chromosome. Okay, when they match up or line up in prophase 1, okay, that is called synapsis. Okay, which isn't kind of the important part of this. The important part of synapsis is that crossing over could occur, which is when the chromosomes literally cross over or overlap with one another. And that could exchange small segments of genetic material from one chromosome to another and create a recombinant chromosome. That word recombinant means a combination of genetic material, and that's going to be new and unique to the offspring. Okay, so again, kind of relating this back to the broad theme of meiosis. The point of meiosis is to create genetically different haploid sex cells. It's part of sexual reproduction. Okay, so the point of sexual reproduction is to create genetic diversity. Okay, differences in the offspring compared to the parents. Okay. At this point, I'm going to kind of break with the questions. You're welcome to keep sending them in, but I want to make sure that I have enough time to go over with you the 4 state labs that you've done throughout this year that are going to be assessed on the last final section of your Regents exam. Okay, so once you get to Part D, this is going to be the last 13 questions of your exam tomorrow, so you need to stay strong for these. You're going to be asked, what did you do in these 4 state labs? Okay, so I have some images up that's going to remind you of what you were looking at, and I'm going to go through diffusion through a membrane first, okay? This is the one that we did earlier in the year, okay? So this is the one that's going to need the most review, and it had the most going on. Okay, all right, so you can see this. Diffusion through a membrane. We went over, thanks to one of your questions, we went over the definitions of diffusion. It's a form of passive transport, okay? High to low, that's what I mean by concentration gradient. High concentration, my brackets mean concentration, to low, okay, without the use of energy. Okay, so energy use, should I put a question mark here? No, okay. And then we talked about active transport. We did not see active transport in this lab because what we did is we made a model of a cell. It wasn't living, it was just a model, okay? So living cells perform active transport, so we were able to witness diffusion, okay? What you did was you created your cell using a semi-permeable dialysis tube. Inside of your cell, you added glucose and starch. Okay, and what it did was it made the color kind of like a white or colorless liquid. Okay, surrounding the cell, you submerged it in a solution of water and starch indicator solution. What we used was Lugol's iodine. Okay, so this was that yellowish brown color. Okay, we said it was amber. And in your final state, what had happened was the glucose was small enough to diffuse out of the cell. Okay. The starch indicator solution, the Lugol's iodine, was small enough to diffuse into the cell. And the starch was too big. Okay. It remained inside your cell. It couldn't leave. Okay. It was too big, so it stayed inside. Okay. How we identified this was the starch indicator solution interacted with the starch, and it turned the color of the cell a blue or a purple or a black color. And then we used a pipette and collected a little bit of this water surrounding the cell and tested it for glucose, and it tested positive. Okay, we used an indicator that started blue and it turned bright orange when heated up a little bit to show that glucose was small enough, okay, to travel through the cell membrane and leave the cell. Okay, so comes back to size. Okay, small molecules, okay, are able to leave the cell membrane. Okay, polarity means charges, generally non-polar or chargeless molecules readily go into and out of the cell membrane. The second part of this was osmosis. Okay, so my pictures are not in color. Okay, but what we did is we took a red onion cell, right, bright pink, and we looked at it under the microscope, and you were able to see that these cells were kind of rectangular shaped like bricks. You could easily see the cell wall. We can infer that the cell membrane is in there, pushed right up against the cell wall, and the cell was filled with pink cytoplasm. Using staining technique like this, what we did is we added a solution to the slide without removing the cover slip, placing a piece of paper towel next to it so that the existing water could be absorbed through, and then the salt water, okay, could travel across. And it didn't take very long, happened pretty quickly, but what happened was water, through the process of osmosis, okay, was in high concentration inside of the cell, and it moved into the environment surrounding the cell. So that was the saltwater environment. Okay, so what is the molecule that moves during osmosis? Okay, water. So water leaves the cell. So we saw diffusion and osmosis, two forms of passive transport. Your making connections lab, I'm not going to spend too much time talking about because I feel like we went over the importance of a hypothesis, the two experimental variables, the independent and the dependent variable, okay, the importance of a control group and an experimental group. Okay, and how to identify a significant difference and what to do with your data. Most importantly, be able to calculate an average. Okay. In this experiment, you had to squeeze a clothespin. Prior to that, you had to take your resting pulse rate. And then after 1 minute of exercise, notice that your pulse is going to increase after exercise to allow for increased gas movement and things like that within the body. Okay, so designing a controlled experiment was kind of like the focus of this lab. Okay, so that's 2 down. We have 2 labs left to talk about. Okay. All right, this wheel chart is going to show you the beaks of finches. Okay, each student was given a tool that was going to model your beak, and you were given seeds, different size seeds, and we tasked you with obtaining as many seeds as you could in a certain amount of time, and we gave you a cutoff if you did not get enough seeds, okay, due to the characteristics of your beak. Okay, organisms had to migrate or they died off. Okay, so if I zoom in on this Beaks of Finches lab, okay, you're going to be given an example organism. It might be a finch, it might be a different type of organism, but it's always going to have some characteristic, okay, beak structure and size, okay, different factors like the biting tip, things like that, the different edge crushing, okay, that would allow the finch to feed successfully on its food source. And then at the very center, this is kind of like the most important, the type of food. Okay, so organisms that consume mainly plant food, if I were to kind of extend this line out, is going to be everything in this kind of larger semicircle, from the cactus finch up around all the way to the vegetarian finch. Okay. These organisms are going to eat plant food. They're not going to compete with organisms that are going to eat mainly animal food, like the woodpecker finch up to the large tree finch, or something that's going to eat 100% animal food, like the warbler finch. Okay. Relating back to the theme of natural selection and common ancestry, organisms that are most adapted to their environment are going to survive longer. They're going to reproduce more offspring. And so the frequency of these advantageous traits are going to increase in the next generation. Okay. Organisms that show relatedness came from a recent common ancestor. All right, and then last one is the big one, okay? We gave you 4 plants, okay? 1 plant we called Botanicurus. We said that this was the important one, and we gave you other options, and you had to gather first structural data and then something called molecular data in order to determine relatedness. Okay, so structural data is things that you can see, okay, using a microscope or your own eyes, right? You looked at these organisms to identify similarities and differences, okay? Something like a fossil would be a good example of structural data because it could tell us about bone structure, okay? Versus molecular data, okay? DNA and proteins are the best examples of molecular data. And this is the best way to determine relatedness, okay? Sequencing the DNA, looking at proteins and identifying similarities are going to for sure give us some information about common ancestry, okay? So 2 tests that you need to know. Best case scenario, you just need to recognize it as a multiple choice option. Okay. Worst case scenario, you have to pull this phrase out from your brain and write it down on the paper, okay? Paper chromatography, okay? We gave you a piece of filter paper and a couple of bottles of the plant extract, which started off green, okay? You placed a drop of each of the plant extracts on a line, and then you placed this filter paper in a small beaker of water, and the water line had to touch the filter paper, but it had to be below the pigment spot, okay? Because the point is that the water will be absorbed up and through the pigment spot. And what you'll get are all of these variations in pigments. Pigments means colors. So, okay. Comparing this to the process of gel electrophoresis, okay, being able to look at a diagram such as the one right here, okay, of an unknown species, okay. Each of these lines we call a band, okay, a band in the gel. And it kind of represents the barcode. It's unique to each of these species. And by kind of like drawing a dotted line over, and being able to identify what bands are in common from the unknown species to species that are sequenced next to it, you'll be able to count the number that's in common and identify which one has the highest number. It's going to be more closely related. Okay, it's going to have a common ancestor, more in common. Okay, so again, that process is called gel electrophoresis. Okay, this is going to okay, size is one thing that's commonly asked when we talked about this process of gel electrophoresis. The other piece could be the electrical current. Okay. DNA is negatively charged, so there's always a negative charge up near the top where the DNA is going to start, and it's going to run towards the positive end. Okay, so once you plug it in, turn it on, the DNA is going to run towards the positive end because of the electrical current that's going through the gel. Okay, smaller fragments are going to be lighter and quicker. Okay, so this right here is the smallest fragment in this gel. Larger fragments are going to be heavier and they're going to drop off a lot closer to the starting point, okay, at the top of the gel. All right, I'm going to have my notes up here from the questions. Any state lab questions that you guys have been getting or anything related to this before we go back to general questions? Nothing state lab, okay, all right. Yep, go ahead.
So when does negative feedback happen, and how does it restore equilibrium?
Okay, negative feedback is a good phrase, and I do want to make a drawing for this for you guys also. Okay, so negative feedback is the process by which we can maintain homeostasis. Okay. Homeostasis is your stable internal environment, okay? Whatever is normal for that organism. So if we're talking about humans, what is normal for us? Okay, one thing would be body temperature, okay? So that is my horizontal line, okay? Body temperature, 37 degrees Celsius, okay? If I'm outside and I'm going for a run, my body temperature is going to increase slightly. Okay, and that's not a bad thing, okay, that's a good thing, right? Because I'm physically active, my body temperature is going to increase. But my body responds to this kind of deviation, so up from normal, by kind of like turning on a negative feedback loop, okay? So there's some sort of threshold, right? It's right here, okay? I'm going to start to sweat. Okay, breathe faster, okay, sweat causes evaporative cooling, and even though I'm outside and I'm still running, it's going to cause my body temperature to drop back down, okay? As soon as I come inside, right, I've got wet clothes because I've been sweating and my skin is wet, okay? Inside the air conditioning, that's going to cause my body to dip down, okay? And so now a different feedback loop is going to be switched on, so I'm not going to be sweating anymore, okay? I get goosebumps, I'm going to start to shiver, things like that, okay? That's going to cause my body temperature to rise back to normal. Okay, so this right here is called dynamic equilibrium. Equilibrium because you want to reach 37 degrees Celsius, that's the goal, it doesn't have to be perfect, okay? And dynamic because it's constantly changing. I'm hot, okay, so I deal with that, right? And then I'm cold and my body responds to that, okay? So the process of negative feedback, it's called a feedback loop, allows the body to maintain homeostasis, okay? So that's a good question, okay? I'm glad I was able to draw that drawing for you guys. Yes?
Next question is from Rose from Brooklyn Technology High School, and she wants to know if you can go over gene therapy.
Okay, yes, yeah. Okay. I don't have a picture to show you, so I'm going to bring back my genetic engineering and gene editing kind of information because we could isolated unwanted genes. Okay, so a gene that could code for undesired, non-functional traits, okay? A protein called restriction enzymes. Okay, are what cut DNA. Okay, so we could cut out a desired gene that's going to code for a functional, or we'll say like a healthy protein, insert it into the human genome, a different enzyme, so not a restriction enzyme. A different enzyme is going to act as a glue, okay, to seal that gene into place. Okay, and once the gene is in the nucleus, it's going to go through the process of protein synthesis, DNA to RNA to amino acids, in order to express that gene in the new host organisms. Okay, so that protein will be created. Okay, so this one right up here could this kind of definition here could be related to. Okay, so different genetic disorders, things like that, okay. Okay, replacing an unwanted gene with a desirable one of the same species for like that kind of common goal. All right, Danielle, you're up.
I have an anonymous question, and they're wondering what role scavengers play in an ecosystem.
Okay, that's a good question. Going back to ecology and the food web, a scavenger is a consumer, okay? So if you think about like that food pyramid, the lowest level is the producers always, okay? Autotrophic nutrition, the process of photosynthesis, or producers, kind of like all of those terms interconnect, okay? That's gonna be the bottom or the basis of the food chain. Okay. As you go up from there, herbivores are going to consume the plants, okay? Omnivores, carnivores from there. A scavenger is an organism that feeds on dead organisms, okay? I would say it's like the opposite from a predator. Okay, a predator is like a wolf or a coyote. It's going to hunt and kill the prey, whereas a scavenger is looking for something that's already dead to prey upon that, food. Okay. All right. So that was a good question related to ecology. Back to the food web. Okay. All right. I see you have more for me. Yeah.
Okay, so we have an anonymous one that asks about the 4 stages of the menstrual cycle, and just to go over them.
Okay. So that is, again, one that's kind of like beyond the scope of what's going to be assessed on the Regents exam. If you remember back to that diagram of the female reproductive system. We talked about important parts like the ovary, the oviduct, the uterus, and the placenta. So the placenta is a temporary organ, right? It's created during pregnancy that's going to deliver the nutrients to the fetus. Well, what if there is no fetus, right? If pregnancy does not occur, okay, fertilization occurs in the oviduct, okay? So sperm does not meet egg, no zygote is created. Then what's going to happen is the egg is not going to implant into the uterus to develop. Okay, the lining of the uterus has been prepared for a potential pregnancy that's going to pass out of the female body along with that egg. Okay, and so that's kind of like the scope of what you need to know for the menstrual cycle. Okay. Back up.
So, what do you need to know about monosaccharides and polysaccharides, and will you really need to know anything on them?
Yeah, okay, thank you. That is a great question. I'm going to go back and I'm going to draw you guys a table on my feedback mechanism sheet, okay, is going to go over the organic molecules compared to their subunits. So the vocabulary that was used in the question was polysaccharide versus monosaccharide. Okay. Vocabulary that's great to know. More simple terms, a polysaccharide is a carbohydrate. Okay, and the carbohydrate is starch. Okay, a monosaccharide is a simple sugar. Okay, and the simple sugar is glucose. Okay, so that is what we need to know, okay? The organic molecule, the big molecule, remember starch was too big to pass through the cell membrane. So when we consume it, it gets digested down into glucose that is small enough to pass through the cell membrane to be transported through the bloodstream. So starch to glucose, okay? Carbohydrate to simple sugar. If I'm going to fill out the rest of this table, I would say next important is proteins. You are a giant protein. Structural proteins are what make up our bodies, okay? And everything that happens in our bodies happens because a specialized class of proteins called enzymes, okay? They're made up of subunits called amino acids, okay? Next important are the nucleic acids. Okay. DNA and RNA, deoxyribonucleic acid and ribonucleic acid. The subunits of DNA and RNA are nucleotides. Okay. And I went over this with somebody's question. Okay. ATCG for DNA and uracil U for RNA. Okay, I'll center this a little bit, okay? All right, the last one, least important. Okay, lipids. Lipids are important. Lipids and proteins make up the cell membrane. It's a lipid bilayer. Okay, what are they composed of? Okay, a glycerol and a few fatty acids. All right, Jaden.
All right, last question I have is what's the difference between animal and plant cells?
Okay, thank you. Okay, so animal and plant cells have several important differences. If we first talk about shape, okay, the shape of an animal cell is generally round, circular, and the shape of a plant cell, if you remember back to that red onion cell that I showed you a picture of, it looked like a brick or rectangular in shape. Okay. Plant cells contain a cell wall and a cell membrane. Okay, and animal cells contain only a cell membrane. Okay, they don't have a cell wall. Okay, the cell wall is rigid and it helps the cell maintain its shape. Okay, another difference between plant and animal cells is the fact that both have mitochondria because they both go through cellular respiration. But a plant cell, in addition, has a chloroplast, which is full of chlorophyll. Okay, chlorophyll is the green pigment found in plants, in a plant cell's chloroplast, and that allows for photosynthesis to take place because the chlorophyll specifically is what's going to absorb the sunlight energy to power photosynthesis. Okay, those are the main differences. There are a few other differences that are a little bit more kind of abstract. They both have vacuoles. A vacuole is an organelle that allows for storage within a cell. Plant cells have one large central vacuole inside of its cell, and that primarily stores water for the plant because water is so important. Okay. Additionally, in the two-cell division processes that we've talked about in this review session, we talk about a little bit the chromosomes. Animal cells have an organelle called centrioles that allow for the movement of chromosomes and help a animal cell divide. But those are kind of like the lesser asked organelles when we talk about animal versus plant, okay? So again, to review kind of like the main ones, the cell wall in the chloroplast in a plant cell, okay, to allow for photosynthesis. Plants also have a cell membrane and mitochondria. Don't let us trick you, okay? They just have those additional organelles in them. I heard the phone ring again, so it's not our last.
No. What is a niche in ecology?
Okay, that's a good one. Okay, so a niche in ecology, or I should say in an ecosystem, is an organism's specific role or their specific job in that ecosystem. And they are very specific. So they are specialized, I guess, adapted to that ecosystem, what they're going to eat, a variety of food sources, okay, where they're going to live, and that is even specialized like along a coastline. If we're talking about kind of like a marine organism, it could be in low tide versus high tide to allow for two different species to fulfill two really similar but different niches to allow for a greater number of organisms and to allow for more biodiversity. Okay, so that's a good vocab word, all right, to be able to see and in your brain think an organism's job or what an organism does in the environment. Okay.
What is the difference between meiosis and mitosis?
Okay, so that's one that we have covered, and I'll go over kind of the more broad definitions. Mitosis is the production of body cells, taking one diploid body cell and producing two genetically identical diploid body cells through the process of mitosis, and for the purpose of growth and repair. Okay, so mitosis, remember toe, it's part of your body. Okay, meiosis, remember the E, right, stands for sex cells. Sex cells are gametes. Okay, there's more phases in meiosis, but what we need to know is kind of like the general overview of this. One diploid cell produces 4 haploid, genetically different sex cells. Okay, so sperm for males and egg for females. I know, my Twitter people, you have had a break, you're done. Jaden, you've been working on the phones. Yeah. Okay, that was kind of another like repeat question. So looking back, if we kind of, you know, skip over any more repeat questions, I don't think we have anything new. Okay. All right, well, I want to thank you guys for coming to hang out with me on a Tuesday evening to go over all of this content that's going to be assessed tomorrow on the Regents exam. Good luck, okay? It's at noon tomorrow. So I want to thank you one more time. Have a good night, and you guys are going to do great tomorrow. So thank you.